Good morning, dear students. In this lecture, we will talk about Biosphere 2, which is an American Earth Science Research Facility originally constructed between 1987 and 1991 and has been owned by University of Arizona since 2011. The Biosphere 2 is also known as Synthetic Biosphere, and its mission is to serve as a center for research and teaching and lifelong learning about Earth, its living systems, and its place in the universe. So let us know about this Biosphere 2. First of all, a brief overview and a short history about Biosphere 2. Biosphere 2 is a sealed glasshouse complex of size 3.14 acres, and it is located near the small town of Oracle, north of Tucson in desert of Arizona. This Biosphere 2 was completed in 1991. It is a closed ecological system to study about Earth's biosphere. It is named as Biosphere 2 because Earth's natural biosphere is considered as the first biosphere. So it is the second biosphere that is synthetically made. So that's why it is named as Biosphere 2. It was designed by a company named Space Biospheres Ventures (SBV) in 1984, and the purpose was to study process and economic dynamics similar to those of global biosphere. The Biosphere 2 project was launched in 1984 by a businessman, Ed Bass, and a system ecologist, John P. Allen. The idea of construction and operation of this Biosphere 2 was of John P. Allen, who discussed this idea with Ed Bass, a businessman. The total budget of Biosphere 2 was nearly 150 million US dollars until 1991. So, before setting up the main structure of Biosphere 2, a pilot scale or a small scale or a test module of Biosphere 2 was studied between 1988 to 90. This diagram shows the layout of that test module of Biosphere 2, which is small in size compared to the main Biosphere 2 structure. The total area that was occupied by this Biosphere 2 was roughly 480 meter cubes, and it contained a main chamber which was sealed by the space frame with tempered glazing or glass, and it had a system of fan coil unit for the temperature control. So the first structure of Biosphere 2 test module was a space framed chamber with tempered glazing. Inside this chamber, the arrangement of condensation was made uh, by an apparatus, and on the roof of this condensation apparatus, vegetations were grown. So the second arrangement, you can see the uh, growing of vegetation on the roof of this condensation apparatus. Then it had a fan coil unit for temperature control because it was covered with the glass. So if temperature is not controlled, then inside temperature would rise up, leading to having difficulty for the plants and other animals to survive inside the chamber. So temperature was controlled by the equipments. Then next arrangement was setting up analytical sensors and data collection equipments. So different type of data was regularly monitored, such as humidity, temperature, flow of wind, then change in temperature from day to night. So like this, many other information was collected using these analytical sensors throughout the experiment. It had arrangement for human habitat also. So uh, human beings were allowed to stay inside this closed sealed chamber for 21 days during the experimental time, and the arrangement for human habitat was human quarters with bed, desk, computer and video. The next arrangement was food and oxygen producing plants that also utilize carbon dioxide, because plants, animals and human beings were retained inside the closed chamber, so there would definitely be release of carbon dioxide because of respiration process. So in order to utilize the carbon dioxide uh, and produce oxygen in return, the plants were also put inside this closed chamber as it happens in the natural biosphere. So the internal conditions were maintained similar to what happens in the natural biosphere of Earth. So there was arrangement for supply of oxygen as well as utilization of carbon dioxide. Then this main structure was connected to a floating to a structure having a floating dome, which they call as floating lung pan. The name of this structure was uh, the lung. The purpose of connecting this structure with st- floating pan to the main chamber of test module of Biosphere 2 was to maintain the amount of air inside the Biosphere 2, because during the daytime, when sunlight used to enter inside the chamber through the glazed glass, the air that was present between the air that was present inside the closed chamber used to get expanded. So because of thermal variation, temperature variation, there was variation in the pressure of air. So in order to balance that pressure, the main structure was connected to another structure which they called lungs, so as to reduce the pressure shock because of expansion and contraction of gases. So there was arrangement for balancing the pressure inside the biosphere test module by connecting it to a floating uh, lung pan structure which they called as lungs. So the lungs were connected to the main structure with underground lung tunnel. There was also arrangement for shower, sink and toilets which were connected to the water recycling system. So all the wastewater that used to get generated during the various activities inside this biosphere test module, it was recycled using a natural purification of wastewater. So like this, the Biosphere 2 test module was designed and operated between 1988 and 1990. The human beings were also allowed to stay inside this closed chamber for 21 days so as to see whether mankind can survive in such closed natural units or not. The main features of Biosphere 2 test module were as follows. The main structure of this test module was 7 meter by 8 meter in size. The total volume was 480 meter cube. It allowed sunlight through a laminated glass, as I discussed before, and a steel space frame superstructure. And it was sealed underground with the stainless steel liner. The internal environment of the Biosphere 2 test module was kept completely isolated from the rest components of the natural biosphere of the Earth. Even The base of the Biosphere 2 test module was lined with the stainless steel layer so as to isolate it from the soil of Earth. The There was arrangement for installation of a movable volume chamber, which is called lung, which was shown in the previous slide. And the purpose of this lung structure or movable volume chamber was to absorb the volume changes due to dangerous pressures from expansion or contraction of air inside the tightly sealed structure. So, in order to avoid the pressure shocks, the main structure was connected to a movable volume chamber, which was called lung chamber. Selected plant species from tropical ecosystems were grown inside the test module. This was perhaps the first experiment worldwide to keep a human inside a system designed for bioregeneration of water, waste, food, and air driven by natural sunlight for 21 days. So, human beings were kept inside for 21 days. An air purification system using a soil bed reactor was also developed to remove the trace gas contaminants such as ethylene, carbon monoxide, and methane from outgassing of structures, material, plants, and humans. Because it was a closed structure, so the trace gas contaminants, which were identified as ethylene, carbon monoxide, and methane, were these these gases were removed using a air purification system that was installed inside this Biosphere 2, because these gases could put adverse impact on the living organisms such as plants, animals, and human beings inside the chamber. So it was necessary to remove them. So what was done in that? Uh, Soil bed was prepared, and the entire air of the sealed structure was used to pass through this soil bed with a pressure pump. So whenever such process was carried out, the trace gas contaminants used to get filtered through the soil bed reactors, and the inside air thus becomes free of these polluted gases. So these were the main features of Biosphere 2 test modules. Now, after getting results from the Biosphere 2 test module, it was seen that now this Biosphere 2 can be scaled up to a large area. So the Biosphere 2 field module was set up. The total area of this Biosphere 2 structure was 3.12 acres, and it was designed to accommodate a team of 8 people who would live inside it for 2 years. A team of 8 people were kept inside the closed Biosphere 2 of size 3.12 acres, and they were kept inside for 2 years. And they were instructed to obtain all their food by growing crops or by eating domesticated animals, replenishing the oxygen by plant photosynthesis, and recycling of all their consumables. Five different types of biomes were established inside this structure, which we'll discuss in the next slides. So, in this slide, this picture shows the image of Biosphere 2 structure, image of main Biosphere 2 structure. You can see the entire structure is covered with the glass and a space frame structure, and inside this structure, five different biomes were established. What were these biomes? We'll discuss later on. So this picture shows the team of Biosphere 2. These are 8 people who uh, lived inside this structure, closed structure, for 2 years, and they cultivated crops inside this structure for food, and they came out of this structure only after 2 years, once the entire experiment was completed. The building has many innovative features. Its total internal area covers near about 1.28 hectares, that is 3.12 acres, and it has five different biomes, such as rainforest. It has a biome which is agroforestry, having agricultural crops and tall trees. Then savanna desert, marshy area or marsh biome, ocean with the coral reef, and a human habitat zone with all creature comforts such as kitchen, library, dormitories, etc. This image shows the agroforestry zone of Biosphere 2, having agricultural crops as well as the tall trees of the forest. The below image shows the intensive agriculture practices in the agroforestry zone inside the Biosphere 2. You can see a number of crops are being grown inside the Biosphere 2 structure in agroforestry zone. The Biosphere 2 is fully automated, equipped with computer controlled internal weather systems and recycling systems. The all the natural process that occur in the natural biosphere of Earth were controlled or maintained using IT based technologies inside this biosphere too. The mechanical systems were installed to generate tides, wind, waves, and rain. Uh, Heating, cooling, and electricity were also supplied from an energy center external to the structure with solar panels to supply much of the energy. So all the energy required for heating, cooling, and electricity were supplied using solar panels, and these solar panels were installed outside the structure. The water is conserved inside the biosphere to mesocosm, uh, which were desert, marsh, savanna, and rainforest. And this water was recycled by condensation by cooling coils. So whatever water gets evaporated, it was condensed using the cooling coils and sent back on the ground. And there was arrangement for the artificial rain or irrigation using sprinkler systems, and evapotranspiration and subsoil drainage were also controlled using the equipments. So these pictures shows the inside facilities of Biosphere 2. The above picture shows the biome, that was the desert biome, and the below picture shows the specimen of coral collected from this ocean biome inside the Biosphere 2. The plant species were selected on the basis of animal food requirements. Inside this Biosphere 2, near about 3,000 varieties of plants and animals were kept. The plant species which were grown inside the Biosphere 2, they were selected on the basis of animal food requirements, the food requirements of animals which were kept inside the Biosphere 2. For example, the African bush baby was also kept in Biosphere 2, and it requires fruits and acacia gum. So the plants providing fruits and acacia gum were also included in the Biosphere 2. The flowers of family Compositae were also introduced to support the bees, because bees were also present inside this closed structure. So to support them, the flowers of Compositae family were introduced. They were also included. Near about 3,000 species of plants and animals were introduced in Biosphere 2 in 2 years. For building invertebrate and microbiota diversity and biomass, which are part of the natural biospheres, for maintaining the invertebrate population and micro organism biodiversity, and also the microbiomass. The soil was inoculated with the natural soil cores and mycorrhiza. The water was imported from the natural ecosystems. For example, for constructing the ocean ecosystem inside the Biosphere 2, the ocean water was taken from the nearby ocean and it was kept inside the Biosphere 2. The flying insects which are attracted to the light were also introduced. So each and every precaution was taken so as to maintain the conditions similar to the natural biosphere of Earth. The local limestone was used for lining the base of marine and marsh ecosystems inside the biosphere too. So this is one picture which shows the animal cages inside the biosphere 2 in 1992. Now the experimental results of biosphere 2. The entire experimental period collected lot of data related to the different activities or processes which happened inside this closed structure. And the major findings of the experiments that carried out in Biosphere 2 are as follows. The mean residence time for CO2 in Biosphere 2 was monitored between 1 to 4 days. Whereas in the Earth's biosphere, this mean residence time of carbon dioxide is near about 3 years. The ratio of carbon in the living biomass means microorganisms, plants, animals, to the atmospheric carbon was 100 is to 1. This means compared to the atmospheric carbon, the carbon in the living biomass was 100 times more in biosphere 2, whereas this ratio in the Earth's biosphere is 1 is to 1. So a huge variation was seen in the ratio of carbon in the living biomass to atmospheric carbon in the biosphere 2. Then the next parameter was ratio of soil organic carbon to the atmospheric carbon. Soil organic carbon means organic carbon that is present in the soil with respect to the atmospheric carbon. The ratio was 5000 is to 1, whereas it was 2 is to 1 in the Earth's biosphere. So a huge variation was seen in the composition of carbon with respect to the atmospheric carbon in biosphere 2. Also talking about the oxygen concentration, the Earth's biosphere has an oxygen concentration of 20.94%. All of us know this. In biosphere 2, the oxygen concentration was dropped to 14.5% by the middle of Jan 1993. And during that time, seeing the urgency, the oxygen from outside was supplied for several weeks so as to increase the concentration of oxygen in the biosphere 2. The oxygen concentration dropped within first 4 months of the operation of this biosphere 2 very rapidly. So these were the experimental results of the biosphere 2, and it was the results were compared with the Earth's biosphere, and the experiments run till 1993. The team of 8 people was kept inside this biosphere 2 for 2 years, and in 2011, this biosphere 2 was handed over to University of Arizona for further research, teaching, and investigations. It is still in operation, and lot of research is being carried out so as to understand the natural processes that occur in the Earth's biosphere, and to see the possibility of establishing such closed biosphere in the other planets such as Mars and uh, others. So, but still there are a lot of future research opportunities which can be taken into consideration while investigating the natural processes inside this biosphere too. Some of these future research opportunities include the reasons for the changes in CO2 and oxygen concentration in biosphere 2, as we saw in the previous slide, compared to the nature's biosphere. The concentration of CO2 and oxygen was found changed to a large extent. Another possibility could be assessment of sensitivity level of biogeochemical cycles in the biosphere too, because lot of biogeochemical cycles are also operating in this closed biosphere too. So, what are the levels of sensitivity of those biogeochemical cycles? Then, understanding the fundamental components and processes of Earth's biosphere and assessing the dynamics of land and water ecosystems in detail. And finally, development of technologies compatible with the sustaining of biospheres. So these are the future research opportunities which are connected with Biosphere 2. And many of these research opportunities have already been carried out through different experiments in this Biosphere 2. So these are some of the references. So that was all about the Biosphere 2, concept of Biosphere 2, which was uh, established in late 1980s and is still in operation. So that was all about today's lecture. Thank you so much.